Hello dear students, welcome to the another video tutorial of mammalian histology. Okay, so in last sessions we have completed with the muscles related to the organ we are studying, that is nothing but the tongue. Okay, we have seen the various type of the muscles, and in today's session we are going to study the histology and the taste bud structure of the tongue. Okay, so the longitudinal mucosa is covered as the stratified squamous epithelium, okay, with varying degree of the keratinization. Okay, the keratinization on the lingual mucosa layer by the stratified squamous epithelium. Okay, and since the dorsal surfaces of oral tongue is more at risk for the desiccation, and abrasions the that going to abrasions form the contact with the food bolus of the varying temperatures and the textures, okay, of the food. It is covered by epithelium that is keratinized, okay, and however the ventral surface of the tongue that as having the more risk of the desiccations with the surface of the tongue, as well as the pharyngeal part that are going to relatively well protected, going to form by the harsh environment. Okay, so these keratinized stratified epithelium on the dorsal surface going to protect the our organ from the harsh environment. Okay, it will be the temperature when we going to eat the hot food. Okay, and um, there will be the therefore the papillae that of the epithelia are going to uh, having the keratinized areas on the dorsal surfaces. Okay, while therefore the epithelia in these areas are non-keratinized also, with having the epithelium is going to adherent, that the underlining that are the stratified muscle fibers, okay, of the tongue. Okay, so there is a fibrous rape that are going to middle the tongue that mask the point of the tongue. Okay, and this is the fibrous rape on the uh, tongue that masks the point of the fusion of the embryonic lateral lingual swellings. Okay, that are the embryonic origins of the tongue development. Okay, so posteriorly there is available or the variable amount of the adipose tissues that are within the pharyngeal tongue. Okay, and that adipose tissues are going to protect for the surfaces. Okay, as you can see in this picture, this will be the dorsal surface of tongue section. Okay, and which is stained with the hematoxylin and eosin stain. Okay, at medium magnification you can see the non-keratinized stratified squamous epithelium for the protection of the organ, okay, from the varying temperatures of the food. Okay, and which are going to you can see the mucosa layer. Okay, you can see the connective tissue papillae, okay, which are going to having the circumvallate into the keratinized epithelium layer. Okay, so having the lamina propria, okay, of the mucosa, and you can see the superior longitudinal muscles of the tongue. Okay, that are going to form the structure for the tongue. Okay, and that are going to um, control the structure as well as the shape and the size of the tongue. Okay, with the transverse muscles of the tongue, as you can see in the picture. Okay, and that vertical muscles of the tongue are going to shorten or going to pull towards the root from the apex. Okay, to control the growth of the that organ. Okay, to control the growth as well as the flexibility and the um, of the tongue to control within the 10 centimeter. Okay, so as you can see, these functioning that coordinating function of the superior longitudinal muscles of the tongue as well as transverse muscles with the ventral muscles. Okay, so these muscles are going to comprises the movements of the tongue within the buccal cavity as well as the modifications that of the tongue going to come function of the tongue going to give smooth function. Okay, so as you can with these muscles are going to have the importance roles into the um, tongue surfaces. Okay, so as you can see, the dorsal mucosa of your um, tongue will be having the keratinized by numerous raised structures known as the lingual papillae. Okay, and they give the characteristic rough appearance to the dorsal surface of your tongue that is not appeared on the ventral surface of the tongue. Okay, so the pharyngeal tongue also has raised uh, dome like structures throughout the mucosa. Okay, so however these are the lymphatic aggregates that are the lingual tonsils. That lymphatic aggregates are nothing but the lingual tonsils and should not be confused with the papillae. Okay, papillae are the different structures than the tonsils. You do not mix tonsils with the um, papillae structures. Okay, so as you can see, the papillae structures, we are the four type. There are the four uh, main type of the lingual papillae are found on the surface of the human tongue. Okay, so these includes, the very first will be the filiform papillae. As you can see, the conical structures arises uh, around the circumvallate papillae in the structure in the picture. Okay, so there are the most abundant of the four type of the papillae. Okay, so these papillae are the most abundant than the four type of the papillae. Okay, so they are the stretched. Okay, as you can see, they are the stretched conical structures. Okay, and their color is gray white papillae structures. Okay, and that color is gray white. Okay, so the papillae that covered in the heavy coat of the keratinized squamous epitheliums. Okay, and these are located on the apex region of your tongue. Okay, so by making the dorsal surface of the tongue that having the rough surfaces, these are the papillae going to provide the friction to allow the movements of the food bolus during the chewing. Okay, so during the chewing, these papillae are going to get communicate with your food and forming the food bolus. Okay, and it should be noted that, so this is very important point. Okay, so this should be noted that the papillae do not possesses the taste buds. Okay, as the keratinized cells on these filiform papillae and going to inform the structures that keratinized structures are not possessing any type of the taste buds on it. Okay, so filiform papillae are not having the taste bud on it. Okay, so this is the important point on in this slide. Okay, so as you can see, the second structure that will be the fungiform papillae are weakly keratinized cells. Okay, and having with the less abundant numbers in the filiform papillae. Okay, however they are scattered across the entire dorsal surfaces of the tongue. Okay, they are scattered in the entire dorsal surface of the tongue. Okay, so as you can see in the highlighted green color patch. Okay, around the keratinized that cells. Okay, these are the structure of the fungiform papillae. Okay, that highlighted in the green color only. Okay, so these are the highly vascular, mushroom shaped, and papillae contains a few taste buds on the apical surfaces of the fungiform papillae. Okay, so as you can see in the third type, that will be the foliate papillae. Okay, that appear as bilaterally paired papillae. Okay, and these are the longitudinal slits. Okay, also known as the longitudinal slits. Okay, and these are the parallel, you can say the paired, okay, bilaterally paired structures, and you can say the posterior margins of this tongue having the with the uh, with location near the succulus terminalis. Okay, and this mucosa is non-keratinized mucosa layer. Okay, and having with the papillae are populated with the numerous taste buds. Okay, so these foliate papillae having the numerous number of the taste buds which are present on the these um, parallel longitudinal slits are also known as the foliate papillae. Okay, so as you can see, the circumvallate, or the it also known as the vallate papillae. Okay, so there are organized linearly. Okay, their organization is linear organization on the tongue. Okay, as a set of 4 to 6 large papillae are inverted, and that going to see on each limb of the succulus terminalis. Okay, that is 8 to 12 papillae in total. Okay, as you can see in this picture, that linear organization, that's small circles with the one single line. Okay, and the dome shape, forming a shape that uh, okay, so these will be the circumvallate papillae. Okay, as circles, patches. Okay, so these will be the 8 to 12 patches in linear number of the having the it location. Okay, so in longitudinal section, the characteristics furrow the found within the papillae can be appreciated. Okay, so these modes that facilitated the drainage, okay, with the salivary vein umbered glands that having the empty into the structures. Okay, so the presence of the lubrication that going to creates the favorable environment for the gustatory particles to dissolve, so that they can be detected by the test buds. Okay, that gustatory particles should be dissolved into the that lubrications to get known to the test buds, to having the which compound they can be detected by means of the test buds if they these gustatory particles are soluble into the these circumvallate papillae secretions. Okay, so as you can see, the test buds, okay, while the taste buds are distributed throughout the entire oral cavity. Okay, and they are the higher concentrations of the tongue. Okay, so each taste bud is clear and having the oral and covered by the stratified squamous epithelium. We will see in the next slide the structure of the taste bud. Okay, so a combination of the elongated test gustatory test supportive cells that going to have, also known as the basal stem cells, um, can be found within the each test buds. Okay, so the gustatory cells that have an apical test pore surrounded by the numerous microvilli, and that binds to dissolve these molecule to bring them to closer into the receptors which are present on these microvilli. Okay, and that receptors which are present on the microvilli are responsible for the taste sensations. Okay, and however these cells have the relatively high turnover rate. Okay, and as they're having the shelf life is about roughly 7 to the 8 days. Okay, and as you can see in the this slide, okay, the picture having the test bud, okay, as I highlighted them with the arrows. Okay, as you can see, they are having on the circumvallate papillae, okay, that having the taste bud locations, okay. As you can see, the ventral sections are detecting with the taste bud locations on the each papillae, okay. So these circuli gustatory cells are nothing but known as the taste buds, okay. And as you can see in the next lying picture, which is residing picture with these taste buds, okay, in the section of the papillae section, which are going to confirm the location of the test buds, okay. As you can see, this is the slide. That section is stained by the hematoxylin and eosin stain, okay. So as we are going to study the histology, we should know this, okay. So gustatory glands, so these are the glands having the main some importance into the gustatory endurance of this particular organ, okay. So as you can see, there are 5 gustatory sensations, okay, that are perceived by individuals, okay. So which 5 gustatory sensations are these are the first will be the sweet, second will be the salty taste, then will be the sour taste, then will be the bitter, and then will be the umami taste, okay. So the microvilli found on the apical surfaces of the taste buds, okay, and these cells are equipped with the various receptors, okay, and that receptors for the various specified compounds, okay, that receptors are specific for each compound on the microvilli, okay. The reaction that generated from the that varying molecules of the various tests, okay, and the reaction which is generated in the compound going to recept, going to capture by means these receptors, okay, by the interactions, okay, and going to give rise to varying that action potential, okay. And when that action potential going to get varying over there, okay, at that time they are going to subsequently pursue these tests, okay. And then your brain is going to study the type of the test, or going to feel that the which type of a test we have tested, and then you are going to know about the test, okay, which test is this sweet, is this salty, or sour, or the bitter, okay. So this saltiness is often associated with the some specific compound known as the cationic compounds, okay. And these compounds that are nothing but the sodium ions, okay. And while the sourness is related to the acidity, okay, that how that concentration of the hydrogen ions is going to form a compound, and that compound is going to get detected by means of the receptors on the various microvilli of the test buds, okay. And each compound having the each separate receptors, okay. So these organic compounds such as the carbohydrate or the amino acids give rise to the sweet taste, okay, while these bitterness is get associated with the long chain organic compounds, okay. And the final test will be the umami, which having the also known as the savory, okay. So is related to compound with the left-handed and that chiral isomers, okay, and that chiral isomers of the glutamic acid, okay. So this will be the as you can see in this picture, the location of the papillae, okay. As you can see, the fungiform papillae, okay, having the keratinized structure on it, okay, and which are going to form going to get interact during the bolus formation, okay. And these fungiform papillae do not possesses the test bud, so this is the important feature, okay. So the foliate papillae as residing to your lateral sides of the tongue, okay, having with the you can say the in the pair structure. Okay, that pairs of the foliate papillae are present on the tongue. Okay, so these foliate papillae are having the numerous numbers of the taste buds on it. Okay, as you can see the circumvallate papillae. Okay, these are also going to possess some type of the taste buds. Okay, so the linear arrangement of the circumvallate papillae, as you can see in the this picture. Okay, so as you can see the another picture on the slides also, so that are going to confirm the circumvallate foliate and the fusiform papillae structures and the location. Okay, so as you can see the B part will be the papillae structures. Okay, how that papillae structures are seen. Okay, that circumvallate structure. Okay, that possessing the test bud on it, the foliate structure, the arrangement of the foliate papillae structures, that then the fungiform the fusiform papillae. Okay, as you can see the last picture, that is nothing but the test bud. Okay, the section of the test bud. Okay, so as you can see the epithelial cells having with the coating on the test buds. Okay, the protective layer. Okay, as you can see the passage, that outer test pore, that passage is covered with the numerous microvilli, and that microvilli having the numerous or the specific structures of the uh, receptors on it. Okay, and that are going to sense by these sensory cells. Okay, which receptor is going to get active, and then that receptor is going to pass the signals to the sensory cells, and that sensory cells then going to sense the test. Okay, which compound is get attached to the that specific type of the um, uh, cells. Okay, and that are going to get support, and that synapses is happen over the synapses cells. Okay, then at the basal cells, the at the nerve, the electric current is generated, and that electric impulses are going to pass by means of these nerves. Okay, and then the brain is going to connect with the test. Okay, so these are the taste bud structures. Okay, with having with the epithelial cells, the outer taste pore, the microvilli, sensory cells, supporting cells to the sensory cells, and the basal cells. Okay, so we are going to stop here, and we have completed with the topic tongue. Okay, so we will meet in the next session. Thank you.